Good morning, I am Rajalingam, biology faculty. I am from Telangana. I completed MSc MD and worked as biology faculty in Karnataka from past 6 years. Today we are going to discussing about biotechnology principles and processes. So biotechnology is actually widely used in all industries like food industry, medicine industry nowadays. So for large production of quantities of any medicines or food. So here what is biotechnology means, we are actually implementing or inserting a specific character in the specific organ like plant or animal. So just to see here, example for that, crop, we need some kind of crop, rice or wheat or in those plants are actually, we need to, cropping is actually resistant to the diseases. Otherwise, those crop is actually growing within the very stress conditions. Those are unfavorable conditions, less amount of water, even though those crop is grown. Because of all those actually characters they are inserted. So this kind of characters. Otherwise, example, rose, yellow, red roses, nowadays we are seeing that yellow color roses, white color roses, all those specific character implemented, inserting within the species. It is actually because of the biotechnology only. Biotechnology involves a specific technology, that is RDA technology. Recombinant DNA technology. So RDNA means recombinant DNA technology. In this technology, there will be different kinds of steps are there. So anyway, here what is the motto of the RDNA technology? We are actually inserting the specific character within the another gene. So this is the major thing. So now first, what is the step? First step of RDNA technology. So isolation of DNA and plasmid. So we need a specific character which is resistant against the disease. Otherwise, it is giving us a different color. So all those characteristics are actually being made by the DNA only. So from where we are isolating things? From the plants. From plant we are isolating. So here all RDNA technology involves number of kinds of enzymes only. So here this plant, from this plant also, if we need to isolate the DNA. Okay now. So which enzyme we are using here? Cellulase enzyme. Cellulase enzyme. Okay now. Otherwise fungi. So from fungi also we are actually extracting or isolating it. Here chitinase. So why cellulase enzyme only means? Cellulose is actually present in the cell wall of this plant cell. Otherwise chitin is present in the cell wall of fungi. So that's why we are using the chitinase. Or if it is a bacteria. So here for this lysozyme. So anyway, lysozyme we are using. So for the extraction of a gene. Otherwise plasmid. So plasmid also we are collecting by the help of this lysozyme only. This is the first step. Isolation of desired DNA and plasmid DNA. After this one, we need to go for the cutting of desired DNA and plasmid DNA. So let us go for the second step. So cutting also by the help of enzymes only. Those enzymes are known as molecular scissors. Molecular scissors are nothing but mostly two types of enzymes only. They are restriction endonuclease and restriction exonuclease. So here restriction endonuclease. So restriction endo and exonuclease, two kinds of enzymes. Those are known as molecular scissors only. Now I want to say one thing. There will be different kinds of enzymes are there. So here now I am discussing two types: restriction endo and exo. Restriction endonuclease touching at the specific site. So here it is a DNA, and this is to form the. So these kind of ends are known as sticky ends. Those are not parallel to each other. This kind of ends are known as sticky ends. Okay now. Otherwise, restriction exonuclease. Exonuclease cutting at both sides of the DNA. See here it is a DNA. Here it is cutting and here it is cutting. Both ends, both sides of the DNA to form the parallel ends. This kind of ends are known as blunt ends. So this is sticky and this is blunt ends. So city and blunt ends. Both are due to the restriction endo and restriction exo nucleus. Commonly there are different kinds of nucleases are there. So all are actually molecular scissors only. First restriction enzyme is into 2. So Hindu 2 is nothing but it is actually extracted from the Haemophilus influenza, and the Roman number 2 is actually the serial number within the isolation time within the laboratory. Okay, now so sequence of isolation. So this is actually one kind of another kind of restriction enzyme also there. It is EcoR1. So Escherichia coli, and R is nothing but this is actually strain name, and Roman number 1 indicated in the isolation time. So there are different kinds of restriction enzymes. All are known as molecular scissors only. But first restriction enzyme is Hindu 2, Haemophilus influenza. Okay, now so here plasmid. So we need to understand the plasmid also. So plasmid is actually double stranded structure, and it is circular. Okay, now this is also known as extra chromosomal genetic material. So here it is containing number of restriction sites and cloning sites also. That sites are also known as multiple cloning sites. MQS, multiple cloning sites. So here what are the multiple cloning sites? So here you see. So these are all of the different kinds of restriction sites and cloning sites. See here, ampicillin resistant gene and tetracycline resistant gene in this one. These two are known as selectable markers also. Okay, now, because we are identifying the recombinant DNA and non-recombinant DNA by the help of these two genes only. That's why they are also known as selectable markers. See here, this is EcoR1, CIA1 and HIND3 and Biotechnology and PSR1 and PDU2. So here you see ROP, repressor of primer. Repressor of primer is nothing but it is actually regulating the copy number of a proteins. So if we need the 100 plasmids only, that 100 only will become more than 100 never comes. At the same time, ORI, where the replication starts, where the cutting starts, that is known as ORI, origin of replication. So let us discuss about the function of this tetracycline resistant gene. Commonly, we are identified as these selectable markers only during the selection of recombinant DNA, how they are actually giving as a selectable markers. See here, when we are actually removing the ampicillin resistant gene. Okay, now here no ampicillin resistant gene. This case I took the another one, foreign DNA I implemented. So what is happening? There will be loss of ampicillin resistant within the plasmid. So this is actually known as actually insertional inactivation. When the inserting of foreign DNA, then there will be inactivation. This will be not having the resistant against the ampicillin gene only. If I inserted here resistant means it is inactivated. So this process is known as insertional inactivation. So when the new colonies are formed, in this one, there are mostly 3 types of colonies are formed. Here, there is no RDNA. RDNA is substandard. This is known as non-conformants. Non-conformants. These two, which is having the RDNA. This is RDNA. They are known as conformants. Okay, so transformants to non-conformants. Out of these two, okay, now, if I actually kept into a medium, tetracycline medium, okay, now, tetracycline medium, this two is actually functional. This two is functional. But if I take into another medium known as ampicillin medium, Okay, now here one bacteria is completely suppressed, another bacteria is active. So which is suppressed within the ampicillin gene medium. This suppressed bacteria only containing the rDNA. So here it is the rDNA containing bacteria. It is actually inactive, suppressed, and it is active, but it is not a recombinant. So here transformation only two types. They are recombinants. Recombinants and non-recombinant. Two types of transformants, recombinants and non-recombinants. Okay, now when we are inserting into the tetracycline medium, both are active. But when we are inserting into the ampicillin medium, one will be suppressed. Which is suppressed? That is only recombinant DNA. Likewise, we are finding by the help of this gene. So that's why it is known as selectable marker. Okay, now anyway, we are actually cutting. So third process, joining of common desired DNA and plasmid DNA. So here I am joining with a desired DNA and plasmid DNA. So plasmid yellow color one is indicating plasmid DNA. This one completely desired DNA. Now it is rDNA. It is rDNA. rDNA is formed. So what is the after the joining means? What is the major enzyme for the joining of this DNA means? We are using the ligase. Ligase enzyme is also using for the joining of the two DNAs, plasmid DNA and desired DNA. So now rDNA is formed. So here what is the next step? rDNA formation. So here inserting. So inserting of the rDNA into host cell. So which is using as a host always Escherichia coli. E. coli. Okay, now Escherichia coli actually we are using as a host. So when I am inserting into a host, this one inserting into a host, bacterial DNA and plasmid. So I am inserting. After 20 minutes, it will become. It is actually divided into two cells. For every 20 minutes only, Escherichia coli will go divide. But commonly 18 minutes requires for the replication of DNA. So this is commonly takes place in the after the insertion. So here this plasmid is artificial plasmid. Artificial plasmid is nothing but PBr 322. PBr 322. So PBr is nothing but plasmid is actually discovered by the two scientists, whom they are. One is Bolivar and one is Rodriguez. So that's why this is known as PBr. 322 is nothing but the sequence of isolation. How many times they are actually tried for the isolation. So this is the 322nd time. It is successful. That's why it is known as PBr 322. It is first artificial, but first natural plasmid is PSC 101. So first natural plasmid is PSC 101. So here this is actually about the small introduction about the biotechnology RDNA technology, how the DNA is going to cutting and how the DNA will be joining for the formation of this RDNA. Okay, now after the joining we are going for the gel electrophoresis and there will be PCR also. All we are going within the RDNA technology only. But here after inserting of DNA, what is happening? So another one, obtaining of desired product, and lastly downstream processing. So downstream processing is nothing but separation and isolation of the DNA from the bacteria. So this is known as downstream. In this one we are using also enzymes only, protease enzyme, RNase enzyme for the separation of the proteins and RNAs. Okay, now that is actually separated, and by using the chilled ethanol we are completely purified that DNA. This DNA only now using for the production of antibiotics or food. So this is actually downstream processing. So likewise there will be different kinds of techniques are there. Thank you.